Well, good morning, everyone. Glad you could join us today. I am Philip Roseboom. I am the SDSU IPM coordinator based out of Brookings, South Dakota. And yeah, welcome to Soybean Crop Hour this week. Some things to keep in mind: at the bottom of your Zoom screen should be a chat button as well as a Q&A button. The chat button is where we will post any links that may be mentioned during the presentation. There is some text already in the chat that kind of explains the QA and chat function. The QA button, throughout the presentations today, if you have any question at any time, just click on that Q&A button and put your question into that. And then our presenter, once they're done presenting, will do those questions after that. If you are a certified crop advisor or want a CCA credit, I will display that QR code at the end of today. So after both presentations are concluded, I will share that QR code just for about a minute or so. So have your phone up, the app open, and ready to scan that if you want that QR code or that CCA credit. So with that, my first presenter today is Dr. Madalyn Shires. She is an assistant professor and SDSU Extension Plant Pathology Specialist based out of Brookings, South Dakota. She's going to be updating us on some soybean diseases. So with that, Madalyn, go ahead and start your presentation.
Okay. Good morning, everyone. We're going to be focusing on SCN, charcoal rot, and SDS throughout this presentation today. Like Phil said, my name is Maddie Shires, and Connie Strunk, who is our Extension Plant Pathology Field Specialist, is also on today. So if y'all have any questions, we will hopefully be able to address them. Okay. So the first thing I wanted to bring up in this presentation, I also serve as the director of the SDSU Plant Diagnostic Clinic, and many of you might have used the Plant Diagnostic Clinic before, but we have moved to a new location. We're actually in Berg Ag Hall now. So if you have brought in samples before or mailed them, we have changed addresses and changed locations. We do actually have an overnight drop-off, and that's kind of it's been really nice, especially for SCN samples, because they actually go into an enclosed room now instead of our little box that was prone to blowing away. So anyways, please feel free to reach out to us if you have questions, and take a screenshot of this page if you need our updated clinic info. So the first disease we're going to talk about is soybean cyst nematode. Hopefully most of you have heard of this before, but if you haven't, SCN is the abbreviation for it, and it is the most devastating soybean disease in the United States on a year-to-year basis. Over 30% of yield can be lost in soybeans before foliar problems are actually observed, because this pathogen is a root pathogen. One other thing that we've learned in the last year or so is that we do have nematode resistance developing to our PI A8788 soybean lines, and I'll talk about that here more in just a second. If you've not seen SCN before, the picture that you can see on the screen, I'm not sure if you can see my mouse or not, but these are actually the cysts that are in the blue circles, and the cysts are simply the egg sac for the nematode. So there's 200 to 300 eggs in each of those cysts. That plant had about 60 or 70 cysts that we could easily identify with our eyes. This is the current SCN distribution. We have not tallied for 2023 yet to see if we've added any new counties. The most recent addition was Aurora County in 2022. This year, our SCN numbers have been extremely high. We went from an average of 7,000 or 8,000 in 2022 to an average of 30 or so thousand in 2023, due to a high number of samples that had extremely high egg counts. If you've heard our talk about SCN before, we always advocate that you test for that. SCN testing is free at the South Dakota Plant Diagnostic Clinic through a Soybean Research and Promotion Council grant. So please make sure that you submit samples for SCN testing. If you've never tested for SCN before, it's good to get that testing done to kind of establish a baseline and monitor for SCN presence. It can help you make extremely early management decisions and inform how your crop rotation should be looking to manage the SCN numbers. If you've had a previous SCN detection and it's been more than 5 years since you tested, we are highly recommending that everyone be retesting again. Again, it is free. We, again, are seeing the numbers rising, extremely high amounts, and we really feel like it would be good for everyone to be kind of taking an active step toward management and knowing what their SCN impacts are looking like and determine if management plans that have been made are actually helping reduce the numbers. Another thing that can come with, especially with a previous SCN detection, is we might be able to do HG type testing and tell the producer if what types of soybeans the nematodes are reproducing on in that field. HG type testing is done on samples that have 10,000 eggs per cc of soil or more. So we did have several for 2023 that we are actually HG typing right now. So management of SCN, there's several different options out there. There are resistant cultivars. Again, there's that PI8788, and then there's also peaking. We do highly recommend that you consider rotating these resistance genes. Plant with peaking 1 year, plant with a susceptible the next time, or a non-host crop such as small grains, and then use the different resistance gene the next year. That will help reduce these numbers. Rotate. Again, we are seeing that around 22% of our nematodes are reproducing on that resistance line. So it is something that we're actively monitoring, and we are hoping that everyone else can too. There are some nematicide seed treatments. We have a few examples here, but you can also find those in our IPM guides that we've been planting and handing out at trade shows and at CAT's trainings this year. Management of weeds is also extremely important if you know you have active SCN issues, as that helps keep the numbers down through management of those weeds, since there are some weeds that host SCN. I wanted to take a few minutes and kind of talk about this website here. If you've not seen it before, we just got it launched in October of last year through the SCN Coalition, and it's called the SCN Profit Checker Calculator. So if you've sent in a sample and you have that egg count, you can actually see how that egg count is going to affect your yields and your profit. So this can be found online just by Googling for SCN Profit Checker Calculator. So this is where you'll go to launch that profit checker. We've worked together with multiple states. I believe there's at least 15 states that worked with the SCN Coalition to develop this tool and develop numbers. Whenever you go into the calculator, this is the home page that you'll see, and it kind of tells you what this tool does. And again, this tool is just basically giving you an idea of what SCN might be costing you. So we'll select that we're in South Dakota, or if you're from other states, every state is in there. Then you can enter the egg count per 100 cc's of soil, which is what you're provided through the diagnostic clinic. So in this example, I use 5,500. Then the SCN female index. So that is the number of that's the percentage of nematodes that are reproducing on PI8788, which you heard me mention a few minutes ago is 22%. And this is actually pre-loaded and pre-populated on the website whenever you select South Dakota. And then you can enter in the percentage of sand and the pH of the soil in the field. I just kind of grabbed some numbers here. Then also enter in your expected yield and the projected soybean price. So this is the results that you'll get at the end. It says that with 5,500 eggs per 100 cc's and all of the other factors, our percent SCN yield loss is 6.7, and the estimated profit loss is 90.45, so $90.45 per acre. So I actually went back and pulled one of the highest egg counts from 2023, which was 51,000 eggs per 100 cc's of soil. And I ran it back through the calculator using all of the same numbers. And you can see how dramatically that changed. The yield loss associated with SCN is now 21.4%, and the profit loss is almost $300 per acre. So it's kind of an interesting calculator to look at and kind of understand the impact that SCN can be having on your fields. Now I'm going to move into a couple of other diseases. Charcoal rot, this is one that many of you might have heard about the last couple of years. This disease thrives in drought conditions. This picture here is an example of a severely affected field with charcoal rot. One of the hallmarks of charcoal rot is that the leaves actually remain on the plant after senescence or after death, and you can kind of see that in the picture. The other way to kind of identify charcoal rot is by splitting stems and looking for the black spots that you can see on the right side of the screen in that picture. Those are actually fungal fruiting bodies that are reproducing, and so they will grow both inside the stem of the soybean and outside the stem of the soybean. And those fruiting bodies are what kind of gives the gray or charcoal color that provides the name for this disease. This here is a life cycle of charcoal rot. So we'll start at the top, and that is where well, actually, we'll start on the far left, on the left-hand side. So the fungus is overwintering in soybean residues and in soils as microsclerotia, which are the tiny black fruiting bodies that we see in the plant. As the plant is beginning to germinate and grow, those fungal bodies germinate and start infecting roots. And you can see that at the next stage as we're going clockwise. Then as the season progresses, the fungus colonizes the root and actually begins growing within the stem of the plant and starts blocking the vascular tissue. As the season progresses and we get closer to pod fill or past pod fill, the high amounts of fruiting bodies, both inside and outside the stem, cause a complete blockage of the vascular tissue and will actually start casting that charcoal color. And then the cycle starts over again where these plants that are highly infected are left in the field because they were not able to be harvested, and they overwinter and start the infection cycle again. So the symptoms, which you can kind of see those in that diagram, they are, this disease again is a fungal disease. It is favored by warm summers, warm winters, and drought. So the last few years have been very favorable for disease development. The fungus, again, it overwinters in the soil. It infects the plant through the roots at a very early point in the life cycle. And as I was talking in that life stage diagram, you may not see symptoms of the disease until pod fill is complete because it's very slow to colonize the plant until that point. And it causes plant collapse, and usually around that pod fill time point. It can infect soybean seed, seedlings, and mature plants, but it's most harmful when it infects seedlings. Charcoal rot, if you may have heard about it in corn. Corn is also a host crop. It's not as affected by charcoal rot as soybeans are, but it does serve as a host. So that kind of goes into management. If charcoal rot was a yield loss pathogen for you, you don't want to be rotating with corn because it's not going to reduce disease numbers. It will reproduce on corn and the disease inoculum will build up. So we recommend rotating with a non-host crop such as small grains. There are resistant cultivars to charcoal rot. There are management practices that help reduce the effects of charcoal rot, and that is to conserve as much soil moisture as possible through minimal tillage or no-till, and through lowered seeding rates. There are no fungicides that have been shown to provide consistent control. We have been looking at those. It is suggested in articles here and there that there are some seed treatments, but whenever you kind of dive into the science of it, they actually were not as effective as it was thought they would be. So because of all of these factors and the fact that charcoal rot has been rising in numbers, we are going to do a couple of research trials in 2024. We are hoping to look at what cultivars showed lowered symptoms with charcoal rot in South Dakota weather conditions. So we're looking for a couple of different cultivars and just see how they respond in the field. We're also going to try to look at management strategies, probably residue versus no residue, to see if that reduces the disease severity. We are, again, looking into chemical controls, but so far we haven't been able to identify any that have been shown to be effective. So the final soybean disease that I'm going to talk about is sudden death syndrome, or SDS. This one usually gets a buzz toward the end of the season because of the way that the foliar symptoms look. And if you all see the picture on the slide right now, that is a typical symptom of SDS, but it can also be about 15 other problems on soybeans. It can be nutritional, stress, drought, all sorts of other issues. It looks like other disease symptoms. So foliar symptoms are a little bit hard to be the sole diagnosis of the disease. SDS is a fungal pathogen. It's caused by a fusarium. There are a lot of fungal diseases that are caused by various fusariums. Early in 2023, early in the growing season, we actually had a lot of plants that came in that had fusarium wilt. So that it was not SDS, but it was a different fusarium that was causing a different wilt symptom. So infection by SDS occurs early in the season, especially if the plants are put into cool, wet soils. Symptoms of SDS are going to appear in the July to August time point, usually as that plant is starting to, complete pod fill. Green veins, which is what you saw on the previous slide, and a blue-green mycelia growing at the crown is the most common symptoms that we'll see. The blue-green mycelia is kind of hard to find if the infection level is low, but it is there. This fungus actually infects other bean crops. So if SDS has been confirmed through diagnostic testing to be a big problem in the field, it is important to know those alternate hosts to break the infection cycle through crop rotation. Field conditions for SDS. Fields that do not drain well are more likely to have SDS development. Poor crop nutrition and lowered soil pH makes it more likely for disease development to occur. Also, there's been several studies that have shown that moderate to high soil compaction tends to increase SDS. If SDS has been confirmed to be a problem in your field, there are some tolerant cultivars that can be utilized. Again, planting those warmer and well-drained soils will help reduce the likelihood of that fungus infecting the plant. There are some fungicide seed treatments available. Saltro and Martech 340F are listed on here, but there are some others that are available, and you can find those in the IPM guides. It's also recommended that you try to keep the soil nutrients as balanced as possible to reduce symptom development. Kind of going back to SCN, which I was talking about earlier. It's important to know that SCN and SDS have been shown to interact with each other if they're both present in a field. The diseases do occur independently, so just because you have SDS does not mean you have SCN and vice versa, but it is an indicator that both diseases could be present in the field. There's been several studies that have shown that severe symptoms of SDS have a really high association of there being high SCN presence in the field. It's also kind of been shown in general that SCN tends to exacerbate symptoms of other diseases. So if you're seeing a lot of these other soybean diseases kind of popping up in the field and you're unsure why, and you haven't tested for SCN, could be really good to look into. And with that, I think that is everything I have. Here is my contact information. I just realized I forgot to change my office number. I'm in Berg 261 now if anyone needs to get a hold of me. And here is Connie's information. And that is everything I have.
All right, thank you, Madalyn. It appears we do not have any questions in the Q&A or chat. If any of you have a question right now, please click that QA button at the bottom or the chat and type your question in there, and I'm sure Madalyn will be happy to answer it. I do have a question for you, Madalyn. I kind of asked the same thing of Eric. He presented yesterday, diseases or weeds, but what do you think will be one of the toughest challenges for disease management this coming year? I know it's pretty broad, but
Well, part of the reason we were talking about charcoal rot and SCN this year or for this crop hour is because if we have drought symptoms again this year, if we have if we have another drought this year, both of those pathogens do really well with drought conditions. It's part of why we think that we're seeing the SCN numbers rise. They thrive whenever there's more heat because they can get in more generations per year. So the numbers are just simply higher at the end of the season. So yeah, we suspect that we're going to see more SCN and charcoal rot problems, especially if we do have the persistent heat and drought issues that we've had the last few years.
all right, thank you. It appears as though no one else had questions, so thank you again, Madalyn. That was very informative, and thank you for presenting. Hold on a second. It looks like we just got a question that just came in. They asked, how do I take a proper soil sample to test for SCN? How much soil is needed, and should I take samples from different parts of the field or just by field entrances?
Okay. Okay, hold on one second. My computer is having a problem. So we actually have a sampling guide for SCN samples on our diagnostic clinic website. In general, we recommend that we recommend that you send in I can't remember how many cups of soil it is off the top of my head right now, we have it all on our website, but we do recommend that you take samples from different parts of the field. We recommend kind of doing a zigzag model and taking about 20 core samples from across the field. You can also do targeted sampling, maybe in low areas or sandy areas, since SCN is more likely to be found in a sandy area of the field. I recommend that if you have a large acreage field, that you take multiple samples from across the field and label them so that you know where those came from, just to have a more specific sampling across the field. And if you have SCN, kind of know what area or what quadrant it came from. So yeah, I wouldn't just sample at your field entrances because there's a good chance that you could have SCN in other areas and you'll miss that. So I see a couple in chat. Where do we send the SCN samples? That's where you would send it to our the plant diagnostic clinic. If you'll just look up SDSU Plant Diagnostic Clinic on Google, all of our sampling forms are there. All of the sampling instructions for SCN are also there. But I can also reshare my screen really quick and give you our information. This is our mailing address. So right here, if you need to send in a sample, that is where you would send it to. And then Phil, what was the bottom part of the chat? Something about SDS.
It was where in South Dakota has sudden death syndrome been identified?
It's been mostly found in the eastern part, more toward the southeastern part of South Dakota. I have not had time to put together the distribution map for 2023 yet, but I believe the samples that we got in were more focused toward the southeastern part of the state. So I'll stop my share now. So hopefully anybody that needed this has it. Okay.
All right.
All
Well,
right.
we will thank you for answering those questions. Very good questions. Thanks again for your presentation, all that good information. So with that, we will move on to our next presentation. On the website, it says Dr. Jonathan Kleinjohn is presenting, but he is not here. Obviously, this is Dr. David Karki. He is the SDSU Extension Agronomy Field Specialist based out of Watertown, South Dakota. And he'll be covering John's research, more or less the soybean trials that John carries out throughout the state. So with that, take it away.
Thank you, Phil. Okay, thank you, Phil. And like Phil said, I'm kind of filling in for John, Dr. Jonathan Kleinjohn. He's no more with SDSU, and he took a job with South Dakota Wheat and will be working with Wheat Commission, actually. So he will be kind of indirectly working with SDSU Research and Extension in his new role also. So this will be a quick update on what John and his team has been doing in soybean research in the last few years. So I'll go through what John has in his presentation, and hopefully I can deliver the same message that John wanted to deliver to you folks. Okay, so this is something that says, shows different location in the state. These are the sites that soybean has been tested in crop performance testing program. That's the program that John oversaw in his role in South Dakota. So there are boxes, and then there are black fonts and green fonts and red fonts. So the one in the black fonts are the 5-year averages in those sites, and the bottom one, either it's green or red, those are the 2023 average of those sites. But and if you see red, that means it's the lower than 5-year average that yielded in 2023. And if you see green, the average yield was more than the average 5-year yields in those sites. And if you look at a soybean yield trend, So you can see it's been you know, last 20 years it's been it has a slightly increasing trend if you look at it. And then there's some, you know, good years and bad years, but the trend line is moving in upward direction. And these are the yields from CPT program in SDSU. And quick breakdown of what today's you know, about today's presentation. First, I will be covering about the biological seed treatment in soybeans, and then and the second part of my conversation would be date of planting, and also the fungicide on the top of date of planting. And the last, towards the end of the presentation, will be the planting population, and we'll be sharing some of the results that we've seen in last few years. So here's the biological seed treatments, because we all, you know, talk about biological seed treatments. I'm sure you've heard, you know, in one way or the other. If you haven't heard directly, maybe, you know, somebody you know have heard about biological seed treatments, and some of you may have already been trying these seed treatments in your operations. And here's the definition that came from 2018 Farm Bill. It says a substance or microorganism that, when applied to seeds, plants, or the rhizosphere, stimulates natural processes to enhance or benefit nutrient uptake, nutrient efficiency, tolerance to abiotic stresses, or plant health, that means, or crop quality or yield. So it has a very broad definition, which basically says it will do all the good to your plant, and then obviously, you know, expect more yield towards the end of the season. And another statement that I would like to reiterate here is biological products are not regulated in the United States. And biological products out there can be broadly categorized into two different divisions or categories. One is the biostimulants, that is non-living, primarily stress mitigation. That is, you know, example can be seaweed extracts, humic acid, fulvic acid, enzymes. You have probably heard about biochar. So these are the things will probably enhance some of the activities that's already naturally present in the soil or the plant to uh, stimulate or act as catalyst for the plants and the soils. On the other hand, the microbes or the microorganisms are actual living bodies. Those are isolated from soils, plant residue, manure, etc., and has some relationship like symbiotic fungi or free-living bacteria. And you have the example over there. So there are biostimulants who are non-living, and then the bioorganisms which are living. And but sometimes you kind of collectively hear as biological treatments or application, you know, applicants that you can try it out in your operation, in your fields. So here's a study that's done nationwide. You can see all of the states that's been participating in this study on the orange color and in the map on the right side of my slide. And there are multiple sites across multiple locations and multiple states. In 2022, there are 17 states tried these, and we had about 50 locations in 2023 alone. There are 55 locations in 21 states, and South Dakota had 4 sites, Bath, Brookings, Miller, and Renner, in 2022 and 2023. And the objective of this collective or multi-state project was to identify yield response in soybean to common biological treatments if we see any response to yield. And the second objective was to evaluate the influence on soybean plant nutrient status. Do those make plant healthier? So now okay, my computer's been a little slow. Sorry about that. So here are the products tested in those trials. So there are 9 treatments with 9 different biological products, and then 1 control. So altogether there are 10 treatments, and are looked at or compared with each other in those tests. So results in 2022. So something to note here, only 4 out of the 50 sites across those states reported significant yield differences in yield. And one of them, fortunately, was Renner site in South Dakota, and other 3 were the Wisconsin sites. None of the other states that participated had any kind of significant yield differences on those products tested for the year 2022. And the product, and just looked at the table, that's from Renner, South Dakota. Products were rice, bio yield. So, and then of course the control. So you can see the yield. Hopefully you can see my mouse cursor here. So yield ranged from, control had about 53 and 53.1, and then you have all comparable yields with the biological products. Some were a little slightly more and some were slightly lower. So basically, there are some differences, but the differences wasn't that huge. And South Dakota combined locations for 2022, that means combine all 4 locations. You have control and all other products listed there. Obviously the trial average, if you look at the bottom, row here, my cursor, where my cursor is at, those are the trial averages, and Bath had a probably better weather conditions and a better environment in 2022 to yield more compared to other locations, other 3 locations, Brookings, Miller, and Renner. And if you have combined locations 2023, you again have a trial average of 69 in Bath, and then which others had a little less than that. But if you look at all of the, and one thing I want to mention is if you see a letter behind the number, and if those letters differ from each other, that means they are significantly different or statistically different at 95% confidence level. That's why you have LSD of 0.05. So you can look at those numbers. Those are very, very similar, across all of the locations, and some are probably a little more than the others, but still, they're not really remarkably, high differences in any of the locations. So nationwide results, biological seed treatments, less than 10% of the sites demonstrate a yield response in 2022. So these are the conclusions made from that study, of course. Preliminary results show yield response to be unlikely in 2023, of course, 3 of the 25. And environmental conditions need more study. What that means is, do environment have any effect or interaction with these, live biological organisms that we're trying to put on? And then also, if you would like to do some, try it out in a farm, do it on a limited basis. That means, try it out on a smaller field or smaller piece of land, and then also do some homework, because there are quite a few out there to choose from. So second part of the talk today is date of planting and fungicide treatments on soybean, and and we'll show some results here in coming slides. So 3 planting dates of, I would say, early as April 28th, normal planting, mid-May or May 23rd, and June is kind of later planting. And there are 4 varieties belonging to 4 different maturity groups, 0.7, 1.1, 1.6, and 2.1. And these are all done in Brookings. And the fungicide was Veltime, a 7 ounce per acre at R3 stage, which is like when you see a plant trying to begin to form a pod, that's the R3 stage. And all of these treatments are repeated 3 times. That means 3 replications. So here's how the fields were laid out. So here's the average from Brookings for 2022 test year. So you have, going on the bottom on the horizontal axis, you have 3 planting date, and then for each planting date you have 4 different varieties, all belonging to 0.7, 1.1, 1.6, 2.1 maturity groups. And on the y-axis, on the vertical axis, you have the yields. And you can look at it in the earlier planting date. You have slightly higher yield, in an average about 64 bushel per acre, and the variety of 1.6 maturity group did the best. That was 70 bushel per acre. And the yields kind of flattens out on the other 2 planting dates, although in June 10th, it's lowest yielding, and also the yield was kind of flattened out. There's not much difference in the yield, but the maximum yield difference was observed for the early planting date, and the highest yield was 70, and then lowest was a 61. So there's about 9 bushel difference on the earliest planting date. And here you go, the fungicide on the top of that. So you have yellow as fungicide and blue as no fungicide. for each of those treatments. And you can see, the fungicide treated plots did slightly equal or slightly better for the earliest planting date, and also the May planting date, the first two planting dates. But in the last planting date, there are some untreated ones doing a little better too. Or so it's kind of showing that if you're planting early, maybe fungicide would help. So here are the numbers, fungicide versus no fungicide in all of the planting dates. It actually has some positive response, I should say. For the earliest one, 2.3. The May planting date was 2.5, and the June 10 planting date was only 1 bushel difference, and all of them were positive response. And then 3-year average, you have 61 bushel per acre for the earliest one, 62 for the May planting, and then the June planting was 57 bushel per acre. This was the 3-year averages. And then when you do a 3-year average, you do see some non-treated plots doing a little better than the treated ones too. So there's no real significant difference if you compare each variety, fungicide treated or non-treated. There are barely like a couple bushel difference in an average. And if you average all of them out, there's a 0 bushel difference in the April planting, +3 for the May planting, and then again 0 for the late planting or June 10th planting. So yield by relative maturity, you have the lowest yielding. In an average, altogether, lowest yielding was the 0.7 groups, and then and the kind of increasing trend, but kind of your graph, if you go this way, it kind of flattens out when it reaches 1.6 maturity group. And and so Brookings area where the research was done, maybe the 1.6 was the best maturity group to grow. Results, again, early planting has potential but carries some risk because of the possible late frost in the early season. And consistent response to fungicide R3 of 2 to 3 bushel if planted before June. And full season maturity group, beans maximize yield, but genetics are important. That means you have to choose different genetics or different varieties, and not only rely on just the maturity group. And full season for Brookings location is 1.6 or 1.5. Planting population, that's my last part of the presentation. There are 4 pictures of average plant pulled out from fields that were planted to different seeding populations. The first one was 60,000. Second one was 100,000, 140,000, and 180,000. And there are plant height also in there. So 60,000 has a little bit of shorter height, but kind of bushed out. And 100,000 had 37-inch height. And then height kind of remains in that, mid-30s after that. So there's no much difference in the height, but bushing of the plant does show some difference because 180,000 doesn't look, that bushed out. And soybean does have that tendency to compensate for the lower population. So yield by planting date, you have so you have 60,000 population in the very first bar chart, and then 100,000, 140,000, and 180,000. And then again, 60,000, 100,000 is more than 60,000, 140,000 is more than 60 and 100. And then after that, it kind of, levels out or plateaus. Yeah, your yield, potential or the response to planting population kind of plateaus right there, about 66, and you have 65 per 180K. And this is the average of 2016 and 17. Sorry about that. I'm having difficulty moving my slide. Here you go. So here's the situation with the planting, how it played out, the methods, split plot design, planting date by population by variety, and there are three planting dates: May 1, May 15, and June 2nd, and four varieties again: 1.4, 1.6, 2.3, and 2.4. Three planting populations: 100,000, 140,000, and 180,000 planting populations, and each of those treatments were repeated 3 times in each site. So you have May 1st planting date, May 15, and June 2nd. On an average, May 1 yielded about 72 bushel per acre, May 15th about 64 bushel per acre, and and then June 2nd about 55 bushel per acre. Like in the earlier other tests we did with the fungicide and planting date, as you move towards June, the tendency of the yield tends to be a little lower than the earlier planting dates. So here's a May 1st, and maturity group were 1.4, 1.6, 2.3, and 2.4, and the same maturity group beans were tested in other other planting dates as well. And this is Brookings 2023. You have those maturity group soybeans or varieties were split into 3 different planting populations. You can see the blue ones for 100, 140, and then 180. So 60,000 was not there, just the top 3, because 60,000 already yielded less. So you have 70, green is for 100K, yield a little more for 1.4 maturity group, and same with the 1.6, somehow yield a little more. And again, 180,000 yield a little more, and then 180,000 again yielded, more than the, lesser populations. But if you go to May 15 planting day, and you don't see that trend of having 180,000 yielding consistently more than the others for all soybean varieties. So you have now 140,000 yielding a little more for the 1.4 beans, 1.6 beans the same way, 1.2.3 the same, 2.4 the same, and again you have mixed results in terms of population for the June 2nd planting day. If you look at it, you have 180,000 doing slightly better, just a slight tad bit better than the other 2 planting populations. Again, I'm having some difficulty moving my slide. Here you go. So again, here's Brookings in 2023 yield, average yield for planting dates and the planting population. So you have 76 all averaged. For the 180,000 is little, you have about five bushel difference here for the May 1st planting, and May 15 planting, you don't have that difference, just the 180 doing better, but it's actually the 140 is doing better. And it's very, very, almost incomparable. Almost no difference for the June 2nd planting date right there. So in conclusion, so something to think about or ask a question here after you do something and you try to question yourself and try to learn something new. So is there a planting date by population interaction? That's something, in most cases, 140,000 population is enough. And another thing to keep in mind, if you have a history of white mold, later planting in lesser population could be better, and also in wider rows. Some of you or most of you may already know this. So if you have a white mold history, try to get that air circulation in there, and don't try to compact plant to plant and row to row spacing. If you have a little more of that, it helps with the white mold. So I think that's my last slide, if I'm not mistaken. And here's a statement that we have to show when we give our presentation to our public folks. And here is John Kleinsen's information. And it still has Jonathan Kleinsen SD State email. You can email him if you have any questions. And I would like to acknowledge South Dakota Soybean Council for providing funding for the research that I showed you today. And if you can't get hold of John, here's my information, and I also have my cell phone number in there. So feel free to text, email, or call my office, and we can direct you to the right information and try to answer your questions, whatever questions you may have. That's all I have. I'm handing back to you, Phil, and stop share
All
my
right.
screen.
Thank you, David.
Okay, now I can see some chat. I don't
First
know
thing
if
I'll
you.
mention is hold on. First thing I'll mention, all this stuff, all this data is available online. John does publish all of this publicly. I did post it. It should be the last post in the chat. So if you want to follow that and kind of go through the results more slowly, by yourself, you most certainly can. With that, we do have 2 questions for you, David. What is the row spacing on those population tests? Do you know?
Good question. I don't, but the way I know John's program, it's mostly 30-inch rows. And I
Okay.
think all
And
of his CPT programs is 30-inch rows, and I don't think he did or found different equipment just for this.
yep. And I'm sure if you follow that link, it'll be in those publications as well. Also, are there any numbers for the final stand by the population and dates?
I don't. And John might have had it, but probably he overlooked and didn't put it on his slides.
Okay.
Yeah, we
Okay.
or
And
when I do
again,
something, yeah, I do go out and do the final harvest population or harvest stand. I like to say, but yeah, that was missing there. Yeah, that's a good question. Thank you.
okay. All right. I am going to um share that QR code again to David for covering for John. I think it's real information, that we missed otherwise.
Yep.
So
Thank you.
thank you
Yep.
for that.
Anytime. You're welcome. And thanks everyone for being here this
All
morning.
right.
I know you probably had better things to do, but thanks for, sticking out and, for the presentation. Thank you so much. And we'll see you next week for the small grains.
Yeah, so I'll leave that QR code up. If we want to launch that poll, John, put that up. It's just 2 questions. I believe it helps us create a better program, seeing if what we're showing to you guys is a fit or what you want to see. Some other things to mention. The if you are interested, we will be covering insects. Dr. Adam Veris will be doing that. I'll be covering Adam Veris, my supervisor, so this is something I'm familiar with, but he will be discussing Decty stem borer as well as some other soybean pests that have been problems in soybean. So if you're interested in that, please tomorrow for that. I am going to end the QR code. So hopefully, give you about 10 seconds, and then I'm gonna quit sharing the QR code for CA credit. I'll stop that. All right. It looks like uh John said a link in the chat. Um, all these presentations that are done will be available later on, once we're editing to it. Both available on the website, our extension website through YouTube. So if you want to rewatch any of these presentations, they will be available. CC credits won't be offered on those. First, we can only offer them during these sessions. So that's why I have that QR code up. So it doesn't appear like I have any other questions. Answer that poll for us. There will be a survey going out to our registrants for this event. It's kind of like the poll here. It just helps us refine, our crop hour, make it better in future years here. So with that, thank you all of you for joining us. Hopefully you learned a lot, and maybe we'll see you tomorrow and hopefully next week as well. All right, thank you.